Okay, we are going to start with the biotechnology and its application. What is a biotechnology first of all? We are using a living organisms for a human welfare. So bio means living organisms. We are manipulating inside them. We are taking a cell, we are manipulating something for a desired product. For example, insulin. Insulin earlier was we are getting from horse pancreas and a pig pancreas. But what is the problem? As the population increases, demand for the insulin also increases. So we can't rely on the pancreas of the horses. We killed one horse and we're getting just 25, 30 ml, which is useless. I'm like, obviously, it will be a that one. So we shifted this to the microbes. Can we, scientists has a thought, can we change the DNA of a microbes in such a way that it can be give the insulin? So insulin is basically getting from human. So from the human DNA, one pair or nucleotide sequence is there responsible for insulin is taken off, joined with the vector, introduced into the host cell like a E. coli. Now E. coli act like a why need a vector? Because the human host cell should not host cell should not act like a this is a foreign particle. So vector is needed for that, like plasmid or PBR 322 vector was there. So now, once it incorporated within the DNA of the host E. coli, we can say that now it's able to produce the insulin. So why we choose the microbes? In a small container, we can give the birth of many microbes. And obviously the byproduct is what? Insulin will be there. So by choosing microbes, lesser time, because the multiplication of microbes is very higher. Lesser time, we are getting the production. And obviously utility is also small, na? We don't need a larger spaces. Okay. So biotechnology deals with the technique of using live organism or an enzyme from an organism to produce the products and process of useful to the human. So can I say that antibiotics are nothing but biotechnology only? Yes or no? We can say that. The definition given by the European Federation of Biotechnology is follows. So this definition you have to learn. The integration of the natural science. What is natural science? We are making a curd with the lactobacillus. That's the natural one. I want to make more. So that is the integration will be there. I just enhance the technology will be there. So science and the organism, cells, parts thereof, and molecular analogous. What is molecular analogous? E. coli DNA was not ready to produce a insulin. So we manipulate it. So molecular analogous are there for a production and the services. So let's talk about the principle of biotechnology. First, genetic engineering. What do you mean by genetic engineering? What is engineer? Jugadu persons are there to do the machinery and all. Can we do changes in cell? We can do it with the help of genetic engineering. A technique to alter the chemistry of the genetic material. We are dealing with a DNA. Whatever the we are doing the manipulation into the DNA of the host cell. DNA and the RNA. Why RNA? I know only DNA is responsible for generic material. Why RNA? RNA is also important because RNA act like a primer, right? So RNA has to be there, then DNA can be formed. To the introduce the these into the host organisms and thus change the phenotype. What is phenotype? Appearance of the host organism. Please write this or just make sure you're getting it. Genetic engineering is a in vitro outside the body manipulation of a genes to get a desired products or the character. Please write this in your book. Now, maintenance of the sterile ambience. Why we need a sterile ambience? Now, let me tell you one thing. Cell, it's not easy. It is easy to just say, just take DNA from here, just introduce. It's very difficult. We have to maintain the sterile condition. What do you mean by sterile condition? The moment we try to introduce something, there should not be other microbes should be incorporated inside. Otherwise, undesirable product will be forming, right? So we should to take care of that. So how can we take care? Maintenance of a sterile ambience should be there. Can you see here? A microtubule is there. They want to enter. So what happens if the sterile maintenance is not proper? If you go to the doctor and he want to inject you here, needle has to be new or old. Why? Old is okay, na? Old you can use it. Sterile needle should be there. 2s, because piercing will be there in the skin, other microbes are present on the skin that can go inside, it can create a problem, right? So maintenance has to be there. So in chemical engineering process, to enable growth of the only the desired microbes we want, only desired microbes we want, that is the eukaryotic cell in a large quantities for the manufacture of the biotechnological products like antibiotics. Now if you want antibiotics, there should be not other incorporation, it will be problematic. We want vaccine. What is vaccine? Half attenuated toxin removal substance introduced or a chemical introduced in the body, so body can generate their antibodies response towards it. Now in vaccine should, if the vaccine is contaminated, what is going to happen? What is the purpose of vaccine? Body should be prepared. If the vaccine is contaminated, you are creating a harm to the body, because introduction of the microbes directly you're giving. And enzymes, enzymes should be very properly maintained. This point is very clear to you. Let's go further. Now the techniques of genetic engineering first include the creation of the recombinant DNA. What is the recombinant DNA? Can you see here? Just a picture, but it tells you a lot. Like corn is there, you can change the color of the corn, yeah, you can make it. So we use a rDNA, recombinant. Obviously combination you are hindering and you are giving another combination, so that is a recombinant DNA technology. Use of a gene cloning and gene transfer, use of gene cloning and a gene transfer helps to overcome the limitation of the undesirable gene getting multiplied, although with the desired gene in a traditional hybridization. Traditional method was what? That includes some undesirable product also there. So allow us to isolate and introduce only one or a set of desired gene. You want the E. coli to form insulin, you want E. coli to form other things, it is possible. Rather, he prefer to die. Yes or no? He can prepare only one set of thing at once because his body in such a way, right? He is not complex organism. So within the introduction, undesirable genes into the target organism. Can you see something? Is it possible to give the all corona vaccine together? Vaccine number one, vaccine number two. Can you give it together? No. Vaccine one, after giving the vaccine one, body will respond. You take time. Some had a fever, some had a severe allergy, some had nothing. All possibilities. Vaccine two, everything was smooth because already body has defense mechanism was prepared. Getting my point? Okay. Now let's talk about it. In a chromosome, there is a specific DNA sequence. Yes or no? In chromosome, where are the chromosomes found? Inside the cell nucleus, called the origin of replication. Means what? Means what? If this is a DNA, suppose, okay, particular at a point, it's called ori. Replication will be unwinding will be starting from there. Responsible for initiation replication. Therefore, the multiplication of any alien piece of DNA is an organism. How many times is going to replicate? It depends on the organism. It needs to be a part of chromosome. What do you mean by this, sir? You want insulin should be produced. Are you going to infect every E. coli with your insulin gene? No, only once. Once you incorporate, it undergo the multiplication by its own. And by its own, if it is going undergo the multiplication, definitely is going to number of copies will be generated, and those number of copies will have the same genome will be present with the, so that is called a rDNA recombinant. आपने कुछ छेड़छाड़ की है, अभी वो क्या बना रहा है, फिर से नया नहीं बना रहा है, वही कंटिन्यू कर रहा है. So which has a specific sequence known as the origin of replication. So can't we do in other organism? No. If they don't have origin of the replication, we can't do that. Okay. So we need a specific origin things. Here come the endonucleus, exonucleus, biological seizures. We're going to talk about it. You already done in this chapter, right? So I'm just going to give you the insights. Now, basic steps involved in the process. First, you need isolating the genomic DNA. If I want to synthesis the insulin, I need a insulin gene from the human. Then fragmenting this DNA, we have to fragment it. ऐसे fragment करूंगा, I need a something enzyme should be there. Which enzymes? Restriction endonuclease, DNA polymerase and all ligase. We will talk about that. So fragmenting DNA, then screening. After fragmenting, which fragment is there? On the basis of gel electrophoresis. Gel electrophoresis is the process in which we are screening the fragment, which is a higher molecular weight, will be stay here only. If the electrode is there, anode and cathode is there, electrical charge is there, higher weight, molecular weight is there, remaining. Okay, those who are smaller, they can travel a lot. Okay, so on that basis, we can do screening of the fragment. Then we can do the insertion of DNA into the vector. Why we need a vector? We can't directly infect or we can't directly introduce a gene directly into the host cell, because host cell be responsible for acting like a foreign particle for them. So what's going to happen? They will no use of that. So we need a vector. Which vector? What is the property of vector? It can be plasmid for plant, or it can be a salmonella typhi something is there. Why we are using that? It can be infect very easily. Why we need a vector? The reason behind that, it can be multiply by itself. Okay, along with the host. Introducing the host. Can you skip this? No, we can't skip this. Then culturing the cells, obviously undergo culturation and transformation of the host cell. And this host cell undergo further screening should be there. So now let's talk about first method. The basic steps involved in a process are first, isolating the genomic DNA. Why isolation should be there? I want only insulin, for example. So I want nucleotide sequence in a such a way that going to produce only insulin. I can't take extra, I can't take a less in nucleotide. Okay. So isolation genomic DNA from the donor. From the donor, obviously from where you want. Okay. Fragmenting this DNA into the molecular scissors. Are you going to cut like this? Definitely not. This picture is depicting how the enzymes are working on it. Okay. Let's go further. Third, screening the fragments, desired gene. We have just talk about this. Then insertion the fragment with the desired cloning vector. We need a cloning vector. What is cloning vector? First vector has to be cut open for the position. For example, this is a vector, okay, and this is a desired gene I have attached. They undergo the cloning. They undergo the multiplication. Vector should be multiplied, and one vector we can introduce into the E. coli cell. We can't take a chance only one cell, one cell. We can't do that. Maybe it's not able to survive. Then it will be problematic. Okay. Okay, here is that. Can you see that? It is inserted here in the plasmid, and the red color is what? Your desired one. Now, just one second. Now, what is the next step? What will be the next step? What will be the next step? Culturing this cell to the obtain multiple copies of a clone or desired DNA fragments. Yes or no? Now we are doing copies of vector. That is culturing the cells. Now what is going to happen? If the DNA, if the vector is now compatible, we can introduce into the host cell. So transforming the host cell using this copies to the transfer suitable host cell to as a express. What do you mean by express? Insulin अभी तक बना नहीं है. Insulin is not yet prepared. It will be prepared just after transforming that. So cell become mature, and that time it will be going to produce a insulin over there. This point is clear? Okay, let's go further. Now, can you see something? This is a Salmonella typhimurium. Typhimurium is responsible for formation of typhoid. Okay, it leads to the pathogenic. But if you take out some toxin, it become a plasmid. So in plasmid, gene coding for antibiotic resistance. Why we need antibiotic resistance? Tell me. If for incorporated into the host cell, definitely there are some cells who are acting on it. So it should contain an antibiotic. Every organism secretes something in response or in against the other cell. Okay, that is antibiotic we're talking about. So plasmid should have a antibiotic resistance side, and then it is vector, and then cloning can be done. They successfully link a gene coding antibiotic resistance with the native plasmid of Salmonella typhi. Why they choose Salmonella? Because this has a infective property. So they take a part of that. Are you getting my point? Why am who will consider as a monitor in your classroom? Those who are can balance the academic as well as the other things. The person who is suffering from academic, like not able to perform well in academic, I'm giving one more responsibility on them. It's going to create a problem. It can't able to manage. Those who are already strong in academic, they can mostly become monitor. I'm not undergo the racism, but that is the criteria. Similarly, Salmonella typhimurium plasmid is a more probable, we can say, infection. Okay, so we need as a plasmid, so it can infect the cell directly. Okay, but we are here, we are not infecting the cell, we are transferring. We are giving as a, like a vector. Suppose I'm moving from here, from the door. Okay, the moment I open the door, what going to happen? You also going with me. What going to happen? I have done my active energy, you are passive energy. So getting my point. So vector is just transferring with the passive effect. What is transferring? Desired gene. Okay, please pay attention. Let's go further. Now, recombinant DNA technology, or also called a genetic engineer, deals about the production of a new combination that is called rDNA, right? rDNA of a gene material artificially in the laboratory. Why laboratory? Obviously, laboratory has to be there. This recombinant DNA, rDNA molecules are then introduced into the host cell where they can be propagated and multiplied. Can you see something here? The first thing, okay, let the things are getting here. DNA containing the desired gene, we just take up one. Now what we are doing? Enzymes cut out desired gene from here. Now we introduce into the vector. Can you see that blue color is goes here? Now tell me one thing. DNA vector has to be incorporated from something, some salmonella typhi. So vector should become here and the desired gene. Now they combine. Once they combine, they can transfer into the host cell, E. coli. Now E. coli, can you see the replication is performing? Yes. In a minutes, it can be undergo replication. Yes or no? This process is amplification. This process is called culturing, or you can say amplification of a desired gene. Gene expression will be obviously simultaneously done. Point is clear? Now, basic steps, identification of DNA with a desirable genes. That is, see, again, keep on repeating the same things. But now we go a little bit detail. Introduction of the identified DNA into the host, maintenance of the introduction of DNA in the host, and transfer of the DNA to the its progeny. What is progeny? Give time to progeny, means what? To reproduction. Okay, progeny. Reproduction will be there. So once the reproduction will be there, it will be with the infected, or we can say recombinant DNA. This point is very clear. If you want break, please let me know. Your faces are literally down. So please let me know, because I told you this chapter is very theoretical. Okay. So if you have any doubt, please ask me. Now let's talk about these things. But this is very easy to understand as well. Okay. Because you know what, three steps: isolation, sorry, selection, isolation, and incorporation. But three steps are there, but they in doing this. First, enzyme. Now this is very important. Which enzyme? Restriction. There are two types: restriction endonuclease, restriction exonuclease. Exonuclease are the one, please listen carefully. Exonuclease are the one who is responsible for cutting at the end, 3 dash or 5 dash. We are not using it. We want something to be in between. That is endonuclease. So restriction endonuclease, that is REN, act as a molecular scissors or biological sealer, cuts the DNA at specific sites. DNA ligase, once you cut it open, you need something to make sure to join it again. DNA ligase will be there, helps to join the two DNA molecules. Cloning vectors, what do you mean by cloning vector? Vectors serve as a vehicle to carry a foreign DNA. What is foreign DNA right now? Which is we getting from above into the given host cell. Example, plasmid, cosmid. Plasmid is for plants, cosmid or bacteriophage. Bacteriophage for viruses. Host cell, the cell into which RNA is transferred for a multiplication of desired gene, usually the E. coli. Why we are taking the E. coli advantage? It is not harmful bacteria, that's why we use it. Bioreactors. What is bioreactors? I want to make a food today. I need a utensils. So obviously for culturing purpose, they need a specially designed container used to carry out the biological reaction under the control. Why? What control condition? pH should be maintained, aeration should be maintained, temperature should be maintained properly. These three methods will be there. Tools of recombinant DNA technology. First, restriction enzyme. Which one? Endonucleases. Restriction enzymes belong to the larger class of enzyme called the nucleases. There are two types of nuclease, exonuclease and the endonuclease. Why we choose endonuclease? They make cut at a specific position and that we want. At the end, there is no genetic information is almost there. We want at something should be middle. If you cut at the end, what is the use? No use, because we are not able to, DNA ligase is not going to work on that, right? So, and nucleases, they remove nucleoside at the end of DNA. So not required. We required this one. Now example, what are the example? The first restriction endonuclease, HIND 2. Always cut the DNA molecule at a particular points by recognize a specific sequence. If the specific sequence is there, then only they cut it. By recognizing specific sequence. What is that? GATTC. Between G and A, they cut it. We'll see that. This specific sequence is known as the recognition sequence for the HIND 2. So every HIND 2, HIND 3 is there, different is there. Each restriction endonuclease recognizes a specific palindronic nucleus. What is palindronic nucleoside? If I'm reading from the beginning and I'm reading from the this side also, I will get the same, like Malayalam. Malayalam, if I write in English, if I'm reading from this side or this side, I'm getting the same spelling. No. Okay. Can you see that here? Just one second. What is this code? GAATG. Can you read from here? GAATC. So this is called palindronic nucleotide. Here and here I'm getting the same thing. Okay. So can you read this word? From here, same spelling we are getting. So these are the group of letters. Right now, letters example. We are talking about group of nucleosides that form the same word when read to both forward backward direction. But palindromic sequence kya hai? The palindrome in a DNA is a sequence of the base pair that read same on the two strands when orientated on the reading. Two strands. Now one strand is undergo the, see, DNA is a two stranded. Okay. So one strand is up, one strand is here. So messenger RNA will read his this one. So can I say this one strand is complementary of the next strand? So that's why it's called palindrome sequences there. Okay. Can you see that palindrome? If you're reading from here or there, same sequence is getting over here. Okay. So if you want to cut open, you can cut open between this G and just C, or we can say A till here only. Okay, let's see that what I'm trying to talk about. Okay, this is very important. The enzyme cuts both the DNA strands at the same site. Okay. E. coli. What is E. coli? Escherichia coli restriction endonuclease. Can you see that or how I read that? Okay. Cuts the DNA between the base G and A. From G and A, it can be cut. Okay. So GAATTC is a present in the DNA. So now we have a two DNA, vector DNA and a foreign DNA. I want to incorporate foreign DNA into the vector DNA. What should I do? I will cut. I will keep G as it is. Can you see that? G as it is. I can cut this C as it is. Can you see that? So between what? Between what? G and C. So G and C. Next will be the A. So what is happening here? Just pay attention. I will tell you everything. Sticky end is going to form. If I cut here, okay, and this is cutting here. So obviously what is going to happen? I'm cutting this, okay? This is going to place on each other like this. Complementary base pair. Okay, can you see that? These are the sticky ends. Sticky ends are responsible DNA ligase activity. If there is a blunt end, they can't be more easily incorporated. So we need a sticky ends. Okay, sticky ends, DNA fragment joined at a sticky ends, that is a enhance the activity of DNA ligase. So can you see that same thing is happening here? So along with the vector, we get a foreign DNA. This point is clear. Sticky ends and the blunt end. What is the difference between sticky end and blunt end? Sticky ends has a sticky material. This is very dangerous. Can walk here. Can you see that something? It is very difficult for the blunt end to cut recognition side. But in case of sticky ends, recognition side is very easy. You can directly incorporate. But in kind of uniform edges, no over edge. You can't keep like this over here on the blunt end. But in the sticky end, you can cut little part, you can stick it or not. Stick in order. Okay, so what's going to happen? Okay, this is two ends. Can I stick this on each everyone? Like जैसे पहले था वैसे मैं चिपका सकता हूँ इसको? No, this is blunt end. But sticky end will be exactly going to match everywhere. Are you getting my point? Okay. Now, foreign DNA is come from foreign? No, it's from outside the body. So same restriction and enzymes are required for cut open the DNA, foreign DNA as well as vector DNA. They cut open. This is a specific one. This is what? Oh, I'm so sorry. I have to be pichu. This is a specific DNA I have to incorporate here. Correct? Which enzyme? Ligase will be used. Can you see that? And transform into the E. coli cell for division. You're getting the point? You okay. See, already chapter is done, so it's very easy to understand, right? But this is visualization. नहीं हुआ chapter ये? But you are understanding. Oh, that's great. Then hats off to you. Okay, now start animation. There is no animation at all, but just okay. So have you understood what we are talking about? This chapter is theoretical but very interesting because it's a story. It has a story from start one. So it is very interesting. Other chapter do not have a story like this. Okay. So conventional for a naming of RNA. This I will be skip at for this point. I will skipping this. I will teach you in next class because right now this is not that much important. Okay. I read the this one thing, right? I told you this. E. coli I. E. coli means what? Escherichia coli. RI restriction endonuclease. So from where you get restriction endonuclease? From E. coli. So that is the naming of that thing. Okay, let it be. We can. Okay, I will tell you directly. Nomenclature of the C. Can you see that? E for Escherichia, O for coli, R for strain. R for strain. RY. Strain means what? A scientific name has given to you. Strain means what? A particular species, or we can say that. I for first endonuclease to be discovered. So first nucleus was endonucleus was this. Hint 3. 3 means what? Third endonucleus was covered. Hemophilus influenza. Can you see that? The Hemophilus influenza. Influenza virus is there, right? Okay. So these are the strains. Examples are that. Now let's talk about the gel electrophoresis. Why we need? We have separated the fragments. Now from all the fragments, how you come to know that this fragments I want? On the basis of molecular weight. So you have to run the gel electrophoresis. The cutting of the DNA by the restriction endonuclease result in the fragments of DNA. These fragments can be separated by the technique known as gel electrophoresis. This is agarose gel. The DNA fragments are separated according to their sizes. Separated DNA. Can you see that? This process is called illusion. Illusion means what? See, you can't see the DNA under the microscope. You need a ethylene bromide dye should be added and UV light will be there. And then you can see the bands like this. Okay. If they cover a smaller distance, high molecular weight. If they cover a larger distance, they will be a what? Yes. So separated DNA fragments are visualized by the staining the DNA with a ethidium bromide dye followed by the exposure to UV radiation. Okay. So this is called as illusion, not illusion, illusion. Okay. Now these separated bands of the DNA are cut out by the agarose gel. Gel is there. You can cut open and you can take up the list. So on the basis of molecular weight, we are using agarose gel, gel electrophoresis. These DNA fragments are purified. Can I take this? Can I take this? Yes. And now I undergo the purification I used in the, because ethylene dye was added, na, I need to remove it off. Construction the recombinant DNA with a cloning vector. So from this, what we got? We got a desired fragment of a genome of a, we can say human insulin, for example. So we got it from here. We don't know biological scissors are cutting everywhere. So on the basis of molecular weight only we can understand. This is also undergo the screening. If you're not doing the screening, what's going to happen? Some other protein also can be incorporated. Cloning vectors. Can I say biotechnology is nothing but application is everywhere almost in medicine, in farming, agriculture. Cloning vectors, vehicle for the cloning. What we want? See, can you see that? A bacterium is there. Bacterial chromosome is there. We are not hampering a bacterial chromosome. We're taking a plasmid from it. Okay. Plasmids are a small extra chromosomal. Why we take a plasmid? It has the ability to multiply by its own. So if you do any changes in a plasmid, definitely that can be incorporated. Getting my point? So extra chromosomal double-stranded circular forms of DNA that replicate autonomously. What is autonomously? Without any presence of anything. The term was introduced by this scientist. Can you look at this eyes? Okay. What is the name? Lidberg. Now, here we call about the silent feature of the vector. Let's take a 2-minute break. Okay. Now we're going to talk about the silent features of the vector. A vector should be not reacting, non-reacting towards the desired gene. That is the first point. So it should be contain a origin of replication as per that, okay, so that it able to multiply within the host cell. It should incorporate a selectable marker. Why we need a selectable marker? What is the selectable marker? Antibiotic resistance, ampicillin R or tetracycline. They should be there. Otherwise, what's going to happen? If the screening is done, we can easily remove it off. Getting my point? What I'm trying to say? So they should have resistance. They allow to select those host cells that contain the vector from amongst those which do not. Means they give a liberty to infect the host cell properly. Okay, that it means. So this is ORI from the origin replication will be start. You can't do anything with this. Otherwise, it is a problem. This part you should not do anything because origin of replication is there. Then region into which DNA can be inserted. This is the region. You can cut open here anywhere. This you can't change because antibiotic resistance is there. Okay. Now the vector must also have at least one unique restriction endonuclease recognition site. Out of this, see, ORI you can touch it. Here you can change any changes. Here change. Ampicillin resist. See, either ampicillin or tetracycline will be there. So list one unique restriction endonuclease site will be there to ensure the foreign DNA to insert into it. Yes or no? In a room, at least one window or door should be there. At least air can be exchanged, right? The vector should be relatively small in size. Why small in size? It will go directly into the cell wall. Otherwise, problem हो जाएगा ना वहाँ पे. Yes or no? So this most commonly used vectors are plasmids and the bacteriophage. What is bacteriophage? Those who infect the those who infect the bacteria. So T plasmids contain a tDNA and retrovirus contained by the disarming. Disarming means what is disarm? Disarm matlab arm nahi hai. Means what? Toxins are not there. Okay. In vaccine, can we introduce a normal live organism? No. We need attenuated. Okay. So disarming can be used. This is called bacteriophage. Yes or no? What is this? One second. Cloning vector PUC in your there is PBR 322 is there in your CBSE board. So what is that? They all have something the same thing. Okay, let's go further. I will have the same thing. One second. Okay, you have this PBR 322. What is that PBR 322 is indicating what? Base pair. Okay, number is what? Base pair. So it's a ampicillin resistance is there, E. coli is there, in 2 is also 333, endonuclease 3, PAM HI, SI, sorry, SAII, tetracycline resistance, origin of DNA. So can I say that PBR3 is modified version? It has almost everything. What is the required as a vector? It's fulfilling all the criteria, right? Origin of DNA, see, it's small one, but it's there. But we have a larger site where we can actually, this enzymes can be act. Okay. So can I say PBR is actually the ideal plasmid vector? Yeah. So identification of the recombination. How can you identify? Insertion of the inactivation. The most efficient method of a screening for the presence of recombinant DNA. In plasmid, it undergo the recombinant or not, you have to find it out. You can't directly incorporate. You can't every time microscopic, you can do study. You need some indicators. So here is the indicator that is called screening indicator. What is that? Screening. This is based on the principle that the cloned DNA fragment disrupts the coding sequence of the gene. This is termed as a insertional inactivation. For example, the powerful method of a screening for the presence of recombinant plasmid is referred to as a blue-white selection. What is the blue white selection? If the plasmid undergo the recombinant one, so it's going to produce you the blue coloration. If it is not producing anything, that is a white color. Let's see what is that exactly. See, this method is based upon the insertional inactivation. What is insertional inactivation? Inside something is going. If there is a inactivated, is giving the color. If it is activated, is not giving a color. Okay. So of the lag Z gene, which gene? Lag Z gene present on the vector. The lag Z gene encodes the enzyme beta galactosidase. What is lag Z gene responsible for reading the beta galactosidase, which can cleave a chromogenic substrate into the blue colored product. So if it is present, blue coloration will be there. If this lag Z gene is inactivated by the insertion of the target DNA fragments into it, the development of the blue color will be prevented and give it to white color. What do you mean by this? If it is inactivated, if it is activated, you get a blue color. If it is inactivated, you get a. So what are you doing? By this way, we can differentiate recombinant, that is white color. Because once it's undergo recombinant, there is no lag Z. And if you get a blue color, that means those plasmid are not undergo the recombination. Are you getting my point? So it is not possible every plasmid will go undergo, tell me, undergo recombination. Are you getting my point? So this is blue-white method is used. Can you see that? Some are blue, some are white. Not exactly, but you can identify it. Because this is microscopic way to understand. Why we have to screening the vector? Are we able to, or are we that much fool that we just transfer only vector? We want vector plus recombinant one. So those are recombinant, they give a white. Those who are not recombinant, they can give blue. So competent host, what is competent host? Components or component. Competent host, introduction of a recombinant into the host cell. Once the screening done, we have to incorporate into what? So in RNA technology, the most common method to introduce the RNA into living cell is a transformation during which cell take up the DNA from the surrounding environment. I'm very hungry. Food is front of me. What I will do? I will see here and there and just grab the food. And act like nobody has seen me. But I will eat that food. What is that first method? खाना सामने हम ले लेंगे. Okay. So similarly, क्या कर रहे हैं हम यहाँ पे? RDNA is just, what is RDNA with a vector? Chromosomal is part here. Whose bacteria? They can take up inside as a genetic material. Is it easy now? To understand it is easy, but there are some methods. See, first DNA is hydrophilic. What is hydrophilic? Philic. Phobia means dar, going away. That is phobia. Philic means to love, towards love. Okay. DNA is a hydrophilic molecule. It's love water. It cannot pass through the cell membrane because cell membrane is made up of lipid oil. So simple chemical treatment with the divalent calcium. Divalent means calcium having 2 plus. Ions increase the efficiency. If the calcium is there, now tell me one thing. What is going to happen? Okay. This is your plasmid with the recombinant DNA. It has to go inside the chromosomal. What is going to happen? It's not going inside. Why? It's a hydrophilic. And this layer is a hydrophobic because that is a lipid made up that wants to go inside. So who come into the picture? Calcium coming into the picture. Calcium going to do this open. So what going to happen? One gap is open, so it can go inside. So in presence of calcium, but how? What is a calcium? Divalent, not calcium single one. Okay, divalent should be there. Okay, so one in presence of calcium, it go inside. But if it's not going, then we have to follow second method. What's that? rDNA also called. One second, huh? Something is bothering me. rDNA can also be transferred into the host cell by incubating both on ice. Okay, उनको हमें डराना है. चलो, ice के ऊपर सो जाओ. Okay, what going to happen? What is ice actually? Cold environment. When you talk about the cold environment, what going to happen? If I'm taking ice for a longer period of time in my hand, I'm not getting a sensation. Are you getting my point? There is no sense. So similarly, rDNA also been transferred into the cell by incubating on the ice. Both are on the ice, followed by the placing them briefly at a 42 cold temperature, suddenly heat wave, 42 degrees Celsius. So the membrane will be loosely attached. It can be incorporated directly. Okay, and taken putting them back on the ice, again on the ice. Getting my point? So first will be what? Keeping on ice, senseless, then keeping together, heat wave, it go inside again, ice, then it will be cell wall will be forming. So typical recombinant DNA very easily. And third, micro injection. Can you see the micro injection? But this is very, very delicate technique. Nobody like you need a professional to follow this. Otherwise, what's going to happen? See, in gift or zift, we talk about the in case of insemination transfer, right? You need this micro things will be there. But it is very delicate. A professional is needed. So micro injection method, rDNA is directly injected into the nucleus of cell by using a glass micropipette. But it should be sterile. That is more important. Now, biolistics. What is biolistic? Not lipstick. Okay, biolistics. Gene gun method. Gene gun method. Why? It has been developed to introduce the rDNA into the mainly plant cell by using a gene particle gun. In this method, microscopic particles of the gold and tungsten, because gold and tungsten have a property to penetrate into the plant cell. Cut open कर लेंगे ना? Okay. So gold or tungsten coated with a DNA. So gold, suppose this is a gold. This is a gold foil. Okay. And your rDNA is placed here. And now this is not going to throw like that. A gun will be there. Okay. So can you see the gun? Look at the gun and look at the nucleus. Actually, different thing is there. Okay. Why we're doing this struggle? To introduce rDNA? Yes, rDNA. Vector along with that. Yes, yes. I'm just making you think. Okay, rDNA into the host. We can't introduce directly. It will be create a problem. The last method uses, what is that? Disarm pathogen. That is which bacteria? See, if the pathogen, what is pathogen? Who's creating the problem? Who's creating the cell? You can infect the cell very easily by its own thing, right? So which vector? Agrobacterium tumefaciens, pathogens of a several dicot plants, is able to deliver a piece of DNA known as a tDNA. tDNA is the one who, along with the rDNA, along with the agrobacterium, is going inside the cell. But agrobacterium is not going to form another infection. Otherwise, it will be not suitable technique. This is clear. So the tumor introducing TI plasmid of a agrobacterium now been modified into the cloning vector, which is no more pathogenic. So we have a two vectors right now. One vector combined with rDNA and one vector that is Agarobacterium is helping us to just introduce into the cell. Point is clear? Now, can you see that? Transgenic plant with a plasmid. What is that? Tobacco cell. Why there is a tobacco cell? Can anybody tell me why there is a tobacco cell only? Tobacco mosaic virus. Have you heard about that? Tobacco mosaic virus? The leaf of tobacco contains the nicotine, right? So if the leaf get diseased, they're useless. So we need something. We need to modify it. No other bacteria can be affect the leaf. So tobacco plant cell, we take and transfer cell into what? Can you take what part? Nucleus is taken. Cultured, we get a new plant. Now this plantlet is now transgenic tobacco plant. Where? Can you see that cell wall? What is the cell wall earlier? Can you see? T plasmid is introduction of the what? Can you see the recombinant DNA here? It's creating a cut over there and just transferring it. Once it transferring, can you see that earlier it can be infected nucleus. Now we just take up this and culture. So from one cell, we form a transgenic plant. Getting my point? What we are doing? So this plant is now resistant towards that. Getting my point? Okay. Okay. So have you understood these things? Any doubt? Sir, we are just listening. Just go ahead. Okay. So process. After this lecture, if you are reading now, it will be very easy to understand because this is story-like thing. Okay. Process of recombinant DNA technology. First, rDNA technology involves several steps in a specific sequence, such as again the same thing. Isolation, fragmentation. After the fragmentation, screening will be there. Isolation of DNA fragments. Ligation, DNA ligase will be there. Transferring the recombinant DNA into the host and culturing. Same things. Okay. Isolation of the DNA. Same thing we are doing. Same thing we are doing. Correct. But here something is different. So what is difference? I just want to tell you. Isolation of DNA. DNA should be isolated in a pure form. Now we talk about the fragmentation. Have we talked about which DNA we want? No. So we are talking here that. So hence cell wall can be broken. How can you get a desired gene? In human, you can just incorporate it with DNA? No. You need something technique. So you need a cell first of all. So with a bacterial cell, a plant or animal tissue, enzymes such as a lysozyme. What is lysozyme? Suicidal bats. Cellulose, chitinase, fungus. Okay, it is used. So cell wall can be broken down by these things. Once you cell wall broken down, you can go inside. That is, DNA should be removed from its histone. DNA is the winding one. Histone molecules are responsible for make it wind and like to be in coil-like thing. If you remove histone, unwinding will be there. Once in unwinding, definitely you can now have a sequence of nucleotide. So this can be achieved by using an enzyme ribonuclease. After histone, what is that? Ribonuclease RNA and proteases for a histone protein. Histone protein. Finally, purified DNA precipitate out after the addition of chilled ethanol. उसको भी दारू चाहिए. What is that? Chilled ethanol is required for DNA. Why chilled ethanol? See, first ethanol is what actually? Even you spirit, like doctor is giving injection before that they rub this something. What is that? Ethanol. Don't do this. Okay, what is that? Ethanol is responsible for you're going to pierce. Doctor is going to pierce over here. So that infected area, sorry, that area should not contain any infectious bacteria. If the piercing is done via piercing, it can go inside. So it should be removed. Similar way, whatever the additional material is there, it can be removed off. Even in case of iodine detection on the leaf, you must have seen in the 9th class or 10th class. Okay, let's go here. You're not remembering anything from 9 and 10. Online, okay. Restriction enzyme digestions are performed by the incubating the purified DNA molecule, right? Our chapter is done. Right now, we're just studying the extra thing. Okay, so just okay with. So DNA molecule with the restriction enzyme. DNA is a negatively charged. Now, what is the point? How can you cut open the negative charge thing? Why negative charge? Phosphate groups is there, na? O minus, O minus. P is pentavalent. O minus is attached to O another. Getting my point? Pentavalent is there. So O minus is charged on the DNA. So the positively electrode anode. So hence it moves towards the positively charged electrode. So after having cut the source of DNA as well as the vector DNA with a specific restriction in them. So we are preparing at the one side simultaneously fragments of DNA, desired one. One side we are taking vector and we are cutting vector, and now those both are joining. Same steps, right? Okay. Amplification of the gene of interest using the PCR. Now this is important. What is PCR? Polymeric chain reaction. I have cultured 2 or 3 cells. From one cell, I have millions or billions of copies in a regular particular time period. So I am going to perform the PCR. Polyamorous. What is that polyamorous? What is that? Can anybody tell me what is the full form of PCR? Polymerase chain reaction. Now PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene. See, we have done with all the methods. Now each one method has some different technology like PCR amplification, fragmentation, electrophoresis for understanding the which fragment is there. Now what is the PCR? We need a what? We need a what? Multiple copies of the gene. Whatever the gene. Tell me one thing. Insulin gene, I'm going to cut from your body, your body, your body. No, I'm going to cut from only one cell. Once I get a one gene, gene has to undergo the multiplication because same gene has to be there. Getting my point? So PCR is performed. So PCR is look like this. Wow, look like this. But what is the function of PCR? There are steps in PCR. Region to amplify. This is the DNA. This particular part I want to amplify. So first there is a denaturation. A sudden high temperature will be there. What is denaturation? Sticky ends will be go away. So that is the heat, under the influence of the heat. Okay. Then annealing. What is annealing? Now fragments are done. We are joining it again under the low temperature. After the heat, again low temperature. Sticky ends are not connecting to each other. Primers. So sticky. How can I tell you? Okay. I don't have anything right now to teach you. This is the double stranded DNA. On the heating, sticky ends are removed. These two are there. Can you see that? One and two. One here end, one is overhead. Overhead. So RNA primer come into the picture, they form a strands. From one you get two, from two you get a four, from four, 2L numbers. Okay. So now annealing is done. Now who is responsible? DNA polymerase along with the Taq polymerase. This is called the thermostat. What is thermostat? DNA polymerase is an enzyme in such a way that it can heat resistant. It can be activated in heat resistant. Otherwise, if you take another enzyme, it is heat sensitive to what going to happen? It never work over there. So need a thermostat enzyme that is called DNA polymerase. So can you see that once you cut open these things, now there are two strands. Now primer will be doing their work. And once we get a primer, two strands are formed and this undergo the 30 cycles. So you get a amplified. How many copies? 1 billion copies from one gene. This is PCR technique. So first denaturation under the heating, then annealing, lower temperature again, and the extension with the help of DNA polymerase or Taq polymerase is also called as a thermostat. PCR stands for polymerase chain reaction, synthesized in vitro outside the body by making enzyme DNA polymerase. The process of replication of DNA is repeated many times. The segment of DNA, which is gene of interest, can be amplified to approximately billion times, right? Such repeated amplification is achieved by the use of thermostable DNA polymerase that is called Taq polymerase. Isolated from the bacterium, Thermus aquaticus. The amplified fragment, if desired, can now use to the ligate with the, what is ligation? DNA ligase. So what is the PCR? Polymerase chain reaction. First step, denaturation. Second, annealing. Third, extension. I want confidence. You're saying good. You're saying correct answers, but still. Okay. Now insertion. Now what is the last part? Almost last part. We are about to finish, right? Insertion of recombinant DNA into the host cell. The last part. We are just to incorporate into that. It is very easy to say, but for scientists to do this, a lot of practice, a lot of patience are there. There are several. Tell me one thing. To prepare a coronavirus, how much time we take? Almost 1.5 years. Almost. To do the research because that is the infected one. Nobody can work under that, right? So if a recombinant DNA bearing a gene for a resistant to an antibiotic ampicillin is transferred into E. coli cell, the host cell become the transform into the ampicillin resistant cell. If we spread the transform cell on the agar plate containing ampicillin, only transform. That is blue white wala thing. What we done in blue white. Similarly, this is the thing. We are keeping insertion. What is we are doing? We are checking it is antibiotic resistance or not. So the ampicillin resistant gene is the case is called a selectable mark. I know that right now whatever I'm showing it's going on a bouncer. So I know that. Don't worry. But this is the last part almost. But you understood now what is the method. That is more important. You can see bio is a subject you have to read. Gene of interest, PCR we have performed, right? This is again coming the same things. Bioreactors, the last part. What are the bioreactors? In which or the culturing can be done, right? So bioreactors can be thought of as vessel in which raw material are biologically converted into the specific products. Individual enzymes, etc., using the microbial plant, animal, or human cells in a large scale. So bioreactor provides the optimal condition like maintenance of the pH, temperature, and aeration. Why aeration? Anaerobic condition. Sorry, aerobic condition, not anaerobic condition. Okay. Now simple. See, this is simple. Can you see that? Here is a motor. acid, pH, maintain all these things. They are continuously moving, not very fast. Otherwise, everything will be jumbled up. Slowly, slowly they remove. Okay. It is usually cylindrical or with curve base had to facilitate the mixing of the reactor content. The stirrer facilitate even mixing. Stirrer should be there. What? Even mixing should be there. What happened? Take an example, fish tank. In a fish tank, if you don't use those, what we can say, bubbles, no. Oxygen machine. It is not oxygen, it's the air machine only. Okay. If you're not using, it's not evenly distributed. Yes or no? Similarly, this is happening here. Even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. The bioreactor has an agitator. What is agitator? Continuously agitated. Whatever the form, it should be it get a sedimentation. So it should not get sedimented. It should get suspended. So agitator will be not like this. Why? Speed should be maintained properly. Can you see what is I'm doing? If the speed is high, what is going to happen? These are the living cells. They can be get destroyed. Okay, a temperature control system, pH control system, everything will be there. Now tell me one thing, in fish tank, are you going to change the water? No, obviously yes. What going to happen? If you're going to give a food, those granules, what going to happen with this? Some amount will be eaten, some amount will be mixed. It may be form, algae will be form or something. So we need to clean the water. Even though fish are eating, obviously they're doing potties, right? So what going to happen? Ammonia is getting higher concentration. So we need to regularly remove. Similarly, in this cell also, in this bioreactor also we remove because bacteria is using, so obviously they are waste product also there, right? So we have to remove continuously. Okay, this is the sterile air should be there. Now last part, sprang third tank bioreactor. Why? Can you see that? One agitator is there and air bubbles are there. To give the enhancement, proper aeration, this should be incorporated. So these are the bubbles. Bubbles dramatically increase the oxygen. Getting my point? So they increase the more oxygen. This point is very clear. Just one last slide. Okay, downstream processing. The process includes separation and the purification, which are collectively referred to as downstream. What is downstream? Once the cell grow, desired products are there. So we have to remove desired product and downstream product. What is the waste product is there? So we have covered a second chapter that is biotechnology and its application.